Hey guys, welcome back. In this video, let's try and understand the neuromuscular junction. Neuromuscular junction is usually dealt under these three subheadings. The first, let's understand the definition. Then let's understand the structure of the neuromuscular junction, and at last we will see the neuromuscular transmission. So coming to the definition of the neuromuscular junction, it's very simple. It's just a junction between a motor neuron and a muscle fiber. That's why the neuromuscular junction is also called as the myoneural junction. Now coming to the structure of the neuromuscular junction, there are three parts in the structure. One is called as the presynaptic membrane, second one is called as the synaptic cleft, and the third one is called as the postsynaptic membrane. Let's try and understand each one of them. So first let's try and understand what is this presynaptic membrane and the synaptic cleft. As I have told you in the definition that the neuromuscular junction is a junction between the motor neuron and the muscle fiber. That's why here the motor neuron is going to form the presynaptic membrane. Okay, so let's say this is the motor neuron. Fine, and this is the axon of the motor neuron. So the motor neuron, which usually innervates the skeletal muscle, is a myelinated motor neuron. So this is the myelin sheath which we have. Fine. So this is the myelin sheath of the motor neuron or of the axon. So as the axon is approaching the skeletal muscle fiber, it will no more have this myelin sheath. That means it is going to lose the myelin sheath. Second thing is that the axon terminal is going to have an expanded end, and this expanded portion of the axon terminal is what is called as a synaptic knob, or it is also called as the terminal button. Okay, the third point is that this axon terminal is covered by Schwann cells. Okay, it is covered by the Schwann cells, and these Schwann cells, so this is the covering of the Schwann cell, and these Schwann cells are the one which are called as teloglia. Fine. The next point is that this expanded end, what I have told you as the synaptic knob or the terminal button, that is the one which contains lot of mitochondria. Okay, lot of mitochondria in them. So all these are the mitochondria. Not only that, it also contains lots of vesicles. These are called as the synaptic vesicles. Fine. And these vesicles are having neurotransmitter in them, which is called as acetylcholine. Now let's expand this presynaptic membrane and see what are all the features which we can see. Okay, so let's say this is the expanded portion of the presynaptic membrane. So when we expand the presynaptic membrane, on the inner aspect of the presynaptic membrane, we see some structures, and these structures are called as dense bars. Okay, second thing is that in and around these dense bars, we also notice the presence of what is called as voltage sensitive calcium channels. Okay, voltage sensitive calcium channels. Then we are also seeing just on the inner surface of the membrane, we also see lot of synaptic vesicles. Fine, so these are the synaptic vesicles, and these synaptic vesicles are going to contain acetylcholine as the neurotransmitter. And remember that these synaptic vesicles are clustered in and around the dense bars. Fine, so this is the structure of the presynaptic membrane. Now there is also a postsynaptic membrane here. Fine, this is a postsynaptic membrane. The postsynaptic membrane is nothing but our sarcolemma. So what is the meaning of sarcolemma? Sarcolemma is nothing but the cell membrane of the skeletal muscle fiber. So this gap what we are seeing in between the presynaptic membrane and the postsynaptic membrane, that gap is the one which is called as synaptic. Okay, and this cleft is around 20 to 30 nanometers wide, and it contains a very important enzyme which is called as acetylcholine esterase. So what is the function of this acetylcholine esterase is it helps in the breaking down of the acetylcholine into acetate and choline, and this acetate and choline are reutilized by the axon so that there can be synthesis of acetylcholine again. So in order to just summarize what are all the things which we have understood in the presynaptic membrane is that first thing is that the axon of the presynaptic neuron is going to lose its myelin sheath. The ends of the axons are going to expand, and these are called as either terminal buttons or the synaptic knobs. And these ends are covered by Schwann cells which are called as teloglia. Then one more thing which happens is that this axon is going to invaginate into the uh, skeletal muscle or into the sarcolemma. That invaginated portion of the sarcolemma is called as the synaptic trough or the gutter. Now this expanded end, which is called as the terminal button, it has got a lot of mitochondria and the vesicles. Mitochondria, as we all know, are required for the generation of the energy, and energy is required for the production of the acetylcholine, and the vesicles are the one which is going to contain acetylcholine as the neurotransmitter. On the inner side of the membrane, we also see presence of dense bars, and on either side of the dense bars, we see voltage gated calcium channels. Then we have also seen the structure of the synaptic cleft. What is the synaptic cleft? It's the gap which is present between the presynaptic membrane and the postsynaptic membrane. It is 20 to 30 nanometers wide, and it contains an important enzyme which is called as acetylcholine esterase. So now let's try and understand the postsynaptic membrane. So as I have already told you, like this is the axon, and this is the terminal expanded part of the axon, which is called as the terminal button or the synaptic knob. And this terminal button or the synaptic knob is going to invaginate into the sarcolemma. So this invaginated portion of the sarcolemma is the one which is called as the synaptic trough, okay, or it is also called as the synaptic gutter. Fine. So it is not as simple as it is looking here. So let me draw it again. So if this is the postsynaptic membrane, then this postsynaptic membrane is thrown into folds like this. It is thrown into folds. So when it is thrown into folds, what is going to occur is the surface area of the postsynaptic membrane is going to increase, and these folds are the one which you are seeing here. These are the one which are called as the junctional folds. They are called as the junctional folds, and this part of the fold, this part of the postsynaptic membrane which you are seeing, which is present in between these two folds, that is what is called as a subneural cleft. That is called as the subneural cleft. So now just let me expand this subneural cleft and show to you as to how it looks like. So let's say again, this is the presynaptic membrane, okay, along with the dense bars here, and on either side of the dense bar we have the voltage sensitive calcium channels. And let's say this is the postsynaptic membrane, and this is the cleft which we are seeing here. Okay, this is the subneural cleft. Okay, and here also we have, and here also we have the subneural cleft. why I'm drawing is because if I'm drawing, it becomes easy for you also to draw. You can also draw in your exams. Two important things are present in this subneural cleft. So this is called as the mouth of the subneural cleft. At the mouth of the subneural cleft, we are having what is called as acetylcholine receptors. We are having acetylcholine receptors, and these acetylcholine receptors, these are nothing but ligand gated ion channels. Ligand gated ion channels. So it's an ion channel which will be activated when a ligand comes and binds to it. And what is the ligand here? The ligand is acetylcholine. So when the acetylcholine, which will be released from the presynaptic membrane, comes and binds, there is going to be a conformational change in this ion channel, which leads to opening of the ion channel. Okay, that much you remember. And the type of acetylcholine receptor which is present here is called as nicotinic acetylcholine receptor. We have one more variety which is called as the muscarinic acetylcholine receptor, but in the neuromuscular junction we are having the nicotinic acetylcholine receptor. So where are acetylcholine receptor present? They are present at the mouth of the subneural cleft. So when we go deeper down into the subneural cleft, we see presence of, what is this? This is voltage sensitive sodium channels. These are called as the voltage sensitive sodium channels. That means they are going to open whenever there is a change in the voltage. Okay, and remember that the postsynaptic membrane is also called as motor end plate. So any kind of potential which is developed in the motor end plate is called as the motor end plate potential. So this is regarding the structure of the postsynaptic membrane. Let's just recap what all we have studied. So the postsynaptic membrane is not a straight line. It is invaginated by the terminal button or the synaptic knob. That invaginated portion of the postsynaptic membrane is what is called as synaptic gutter or trough, and there are folds on this postsynaptic membrane, and those are called as the junctional folds. So what happens because of this folds? The surface area of the postsynaptic membrane is going to increase. The portion which is present in between the junctional folds is called as the subneural clefts, and the subneural cleft is having two important things. One is called as the acetylcholine receptors, which is present at the mouth of the subneural cleft, and this is a ligand gated ion channel, and the type of receptor is the nicotinic receptor. Then deeper down in the subneural cleft we have what is called as a voltage sensitive or a voltage gated sodium channels. So these points are extremely important for your exams. Next, let's understand the mechanism of neuromuscular transmission. So what has to happen? What is the basic concept behind the neuromuscular transmission is that whenever an impulse is arriving, okay, in the form of action potential in the presynaptic membrane. So what is the presynaptic membrane? Here the presynaptic membrane is the motor neuron. This impulse from the motor neuron has to get transmitted to the postsynaptic membrane, which is the skeletal muscle, and that is going to cause contraction of the skeletal muscle. So let's say now the action potential has arrived at the presynaptic membrane. Once the action potential comes at the presynaptic membrane, or to be more specific, the axon terminal or the terminal button, what is going to happen? There is going to be depolarization of the presynaptic membrane. Now, once there is depolarization of the presynaptic membrane, this is going to lead to opening of, as I had mentioned already in the structure, these are called as the voltage sensitive or the voltage gated calcium channels. So once there is opening of the voltage gated calcium channels, the calcium from outside is going to enter inside. So this is called as calcium influx. So whenever there is a calcium influx, so we have the synaptic vesicles here. These synaptic vesicles are going to move towards the membrane, and then they are going to release their contents. What do they have? They have a neurotransmitter which is called as acetylcholine. So now they are going to bind with this presynaptic membrane, and by means of exocytosis they are going to release the acetylcholine into the synaptic cleft. Now this acetylcholine which is released into the synaptic cleft is going to come and bind to this receptor. What receptor was this which was present at the mouth of the subneural cleft? This was called as the acetylcholine receptor. And what type of a receptor was this? It was a nicotinic receptor, and it was ligand gated ion channel. So once the acetylcholine binds with the acetylcholine receptor, there is going to be opening of this ligand gated ion channel. So when the ligand gated ion channel opens, what is going to happen is that sodium from outside is going to enter inside. So it is going to cause what is called as the sodium influx. So this influx of the sodium is going to change the potential over the postsynaptic membrane. But this potential is a graded potential, and this potential is what is called as motor end plate potential. Okay, it is called as the motor end plate potential because this change is occurring on the end plate, which is the motor end plate. So that's why this is called as a motor end plate potential. So what is this motor end plate potential? It is a graded potential. So this graded potential will slowly pull the membrane potential of the sarcolemma towards the threshold level. So once it reaches the threshold level, what is going to happen is, next thing is that there is going to be opening of voltage gated or voltage sensitive sodium channels. Okay, so once there is opening of the voltage sensitive sodium channels which are present deeper down in the subneural cleft, there is going to be influx of sodium again, and this influx of sodium is going to cause generation of the action potential in the sarcolemma. So once the action potential is generated in the on the membrane of the skeletal muscle membrane, what is going to happen? There is going to be a rapid propagation of this action potential, and this action potential is also going to reach to a structure which is called as the T tubule. So once it reaches to the T tubule, there is going to be a calcium influx into the muscle, and this is going to trigger the muscle contraction. Understood? So this is how the neuromuscular transmission is. So let me just recap all the steps for you people. The first point is arriving of the action potential at the axon terminal. Second point is depolarization of the presynaptic membrane is going to take place. Once there is depolarization, there is opening of the voltage gated calcium channels which are present in the axon terminal. This is going to cause movement of the calcium from outside to inside, which is called as the calcium influx. Once there is calcium influx, there is movement of the vesicles to the inner side of the presynaptic membrane. This is going to release the acetylcholine from the vesicles by a process which is called as exocytosis. So once acetylcholine is released into the synaptic cleft, the acetylcholine is going to go and bind to the nicotinic acetylcholine receptor. This is going to cause opening of this ion gated ligand gated ion channel, which is going to cause influx of the sodium ions. This causes a rapid local depolarization of the motor end plate, which is called as the end plate potential, and this end plate potential is going to make the membrane potential to move to the firing level. Once it moves to the firing level, there will be opening of the voltage gated sodium channels which are present deeper down in the subneural cleft. Once there is opening of the voltage gated sodium channels, there is generation of the action potential, and this action potential is propagated along the sarcolemma and also T-tubule, which further causes influx of the calcium into the sarcoplasm, and that is going to trigger the muscle contraction. So this is how the neuromuscular transmission is going to take place. Now let's see what is going to happen. Now this acetylcholine has gone and it has bound to the acetylcholine receptor. Is it going to stay there itself, or what is going to happen to this acetylcholine? So remember that each nerve impulse is going to release 60 acetylcholine vesicles, and each vesicle contains 10,000 molecules of acetylcholine neurotransmitter. So you can imagine how many neurotransmitters are released, and acetylcholine is going to stay in the synaptic cleft for a period of only 1 millisecond. Why? What is going to happen? It will be immediately removed by an enzyme which is present in the synaptic cleft, which is called as acetylcholinesterase. Okay, so this acetylcholine will be split into acetate and choline, and these will be taken up again by the presynaptic membrane, and they are again utilized for the synthesis of the acetylcholine. Okay, so why this has to happen? This has to happen so as to prevent the repeated excitation of the muscle. So remember that acetylcholine cannot go and keep on binding to the acetylcholine receptor. If that is going to happen, there is going to be a repeated excitation of the muscle, which is not at all required. Fine. Remember, even at rest, small packets of acetylcholine are released. Okay, even when the presynaptic membrane is not stimulated. So this small packets of acetylcholine which are released, they are called as the quanta of acetylcholine. Okay, and these are going to produce a very small change in the resting membrane potential amounting to 0.5 millivolt in amplitude. So that means even at rest, when I see the potential changes, there is going to be a very small changes occurring in the resting membrane potential amounting to 0.5 millivolts. Okay, this is happening even at rest, remember that. Okay, so this produces a miniature depolarizing spike. Here you are seeing a small spike, and then you see again a small spike, and then a small spike. This is what is called as miniature end plate potential. Okay, so remember this term. Sometimes they will ask you in the exam, what is miniature end plate potential? So it's a potential which is produced even when the presynaptic neuron is not stimulated. Small quanta of acetylcholine is released without even stimulation, that is at rest, and this is going to produce a very small change in the resting membrane potential amounting to 0.5 millivolt in amplitude, and this produces a miniature depolarizing spike, and this is what is called as a miniature end plate potential. Hope this video was helpful for you people in understanding the structure and the transmission, neuromuscular transmission. If that's the case, kindly hit the like button, kindly subscribe to my channel, and share this video as much as possible. Thank you for listening. See you again.